All right, guys, questions for me before we get started here? I did post just homework this afternoon for chapter 12. I'll put together some homework for, and there's also a lecture quiz for chapters 12, 13, and 14, if memory serves. I'll put together some homework for 13 through 16. It's going to be a pretty brief overview of 15 and 16. We'll take care of 13. There's not just a ton of information. 13, so we kind of hit most everything there. And kind of try to get as far through 14 and just a little bit of 15 and 16 tonight. Probably finish up a little bit of 16 on Monday. And a week from today, we'll go ahead and knock out test 4. So that's going to be focus on test 4. Focus will be chapters 12 through 14, and then a little bit of 15 and 16. Okay. And then we just have 2 class periods after that. So including today, we've got 5 class periods. Okay. So we've actually done pretty well for losing almost 3 weeks, being displaced out of the building. We've been pretty good in terms of what we've salvaged. Okay. Test 5, we'll just schedule that at the normal time of the final exam. Okay. It's May 12th. This time, May 12th. We don't have to meet May 10th. Again, I don't mind being available May 10th. If you guys are available and you want to pop in, I'll be here at normal class time and answer questions. I doubt I will have a formal lecture prepared that day, but available if you have some. Okay. All right. With that, let's see here. So we finished up last time talking about the structure of nucleic acids. Specifically, DNA in particular uses adenine and thymine, using 2 bonds to hold those together. Guanine and cytosine using 3 hydrogen bonds to pair those together. So again, hydrogen bonds are relatively weak bonds, but easily formed a lot of times in large organic molecules such as DNA or other nucleic acids. Again, this is review. We saw this last time. Now, one thing, I think I've mentioned this before, but I don't remember, so I thought, you know what, let's just put it in writing on one of the slides. I know your text has mentioned this. You should have seen these terms before. Nucleic acids, nucleotides, if you will, can be classified into 2 categories. You have your purines and you have your pyrimidines. So if you look at their structure here, again, all of them have phosphate group, the sugar that binds to the next phosphate group, and then the next sugar. But then all of them have this nitrogen base, nitrogenous base attached to the sugar in addition to the phosphate. The purines, notice there's 2 ring structures. Those nitrogenous bases have 2 structures on the purines. That's adenine and guanine. Pyrimidines have a single ring structure on that nitrogenous base. All right, so that's thymine, cytosine, uracil, which occurs in RNA. RNA does not have thymine. RNA uses uracil. Those are all single ring nitrogen bases. Now, that's just kind of a technical difference, not something I don't think necessarily we have to focus on, but it's definitely worth mentioning. Okay. But notice that a single ring structure will bond with a double ring structure. A purine will bond with a pyrimidine, and a pyrimidine will bond with a purine. Because they're basically kind of different sizes. So you end up with essentially 3 rings on each rung of the ladder, if you will. Right? So if you end up with an adenine accidentally gets paired with the guanine, they're both too big, and you end up with kind of a bulge in the DNA structure, and that ends up in error. And we'll talk briefly about some mechanisms. There are some mechanisms the cell will try to employ to go back and fix those errors. Okay, but and then again, just quick review. But it's important that we understand that there's two different ends of the of each of these nucleotides, right? So again, we talked about last time. We said, well, this is a five carbon ring. We got 1 prime, 2 prime, 3 prime, 4 prime, and then this elbow here is the 5 prime. The 5 carbon sugar between the phosphate and the nitrogen base on every nucleotide. So the phosphate group on the particular nucleotide binds to the 5 carbon, and then the phosphate group on the next nucleotide in the chain binds to the 3 carbon. And that pattern repeats. And then it flips on the other side of the string. Notice this side of it, because DNA will pair up, it will have 2 sides. And again, because that has to be replicated, that has some advantages to have 2 copies. In case one gets messed up, you have a second one. And also when you're copying it, you can just copy both sides. That's what we'll look at next. But they go in opposite directions, right? You got the 5 prime end down here. See the 5 prime right here. And then at this top is the 3 prime end right here. As opposed to over here on this side of the strand, the 5 prime end is up and the 3 prime end is down. This is 5, 3, 5, 3. So it's important that we understand when we've got 2 nucleic acids that pair up, whether it's 2 strands of DNA, or what we're going to see later is when we're generating RNA, or we've got RNA pairing up with RNA, or whatever the case may be. If 2 nucleic acids pair up, they're going to run in opposite directions. The 5 prime is going to be on top for one strand, the 3 prime will be on top for the other strand. And it's really important when you start matching these strands up for all the processes we're going to talk about today. Okay. All right, so it seems like last time we spent some time kind of laying this groundwork. Where do we go with this today? Well, I have a video that, because it's, I don't know, I like to see it, and if I can do it some kind of animation, that's helpful. And I was like, surely there's some nice animations, maybe just a few minutes. I'm going to pause our recording here. If you're watching from home, just go, I've given you information here how to find it, but I'm going to pause our recording here. All right, so the idea there is we watched this video. Now, I might suggest to you, after you've read the chapter, maybe you already have, if you've been reading through the chapters and you watch this video and you realize that's literally a summary of, in a graphical, auditory form, a summary of about a chapter and a half out of your text. Okay, some of you guys have already read ahead, you know those. What I would suggest to you is after you've had this lecture, after you've read the chapter, whatever order you're doing that in, go back and watch this one more time. I promise you, it's a beautiful, simple, elegant summary. Almost all the high points are in there. It's the reason I gave you this link. I think you'll find that's helpful because it's just kind of repetition. Just like anything else. You repeat it enough times, you go over it, you process the information in a few different formats enough times. I think that's going to be helpful. So again, I thought the graphics there were way better. It's completely accurate, completely on point with what we're talking about in our text. And I thought the graphics there were going to be more helpful probably than anything I'm going to be able to draw. Okay. But again, the basic idea here, yes, Keith, please, thank you. Rough something in here. The basic idea the helicase enzyme splitting the two strands, right? And you have a leading strand where you've got DNA polymerase. We just write a DNA pol for short. I can zoom in a little bit. Okay. And so on the leading strand, the DNA polymerase can just go down the strand and follow, right? Because it can only go in one direction. On the other side, because if the DNA polymerase can go from 5 to 3 in this direction, that means 5 to 3 is this way on this one, okay? And so this helicase is moving in this direction. So what we have to do is on the other side, on the lagging strand, you've got DNA polymerase, and it's moving this way. But what we have to do on this side is we have to break it up into chunks. Another DNA polymerase here. And then on this side, it's already replicated. Right? So as this guy moves on the top, on the leading strand, as this DNA polymerase moves, it replicates as it goes and can just continue and follow the helicase. But on the other side, because the DNA polymerase can only go down the strand in one direction, this one runs, these strands run in opposite directions. So the DNA polymerase has to go this way. But what it does is it just breaks it up a little bit at a time. So you'll have multiple DNA polymerase coming in, laying down a strip, have to move further up the line, lay down a strip, move further up the line, lay down another strip. Okay. And that's as you can see, it's kind of hard to draw, right? But we had to try, and because that helps us process that information and hopefully it sticks. That's reasons like that YouTube video is way better than, as you can see, what I'm going to draw quickly. Okay. Questions here? Does that make sense? It's important, and I'll be honest with you, I will struggle to have memorized. Oh, it goes from 5 to 3, 3 to 5. No, it's important that we understand it can only go in one direction, and as we do it enough, we'll learn. We'll go from 5 to 3. Okay. So, and just kind of some summaries here, a few things. Again, as you've been reading along, you'll notice that we have different types of DNA polymerase. It's not just one. Some of them got different functions. Some of them have different specialties. So again, DNA polymerase is just an enzyme which is going to add nucleotides to a copied or replicated strand. So it's going to add nucleotides as it works its way down the DNA strand. I'm not going to ask you to memorize all of the enzymes associated with these processes, just like we did with the Calvin cycle and just like we did with metabolism in general. There's lots and lots of enzymes. I'm not going to ask you to memorize those here. It's important that we understand that's what's happening. There are lots and lots of versions of DNA polymerase. Now, I did notice, and I don't know how many of you guys have had exposure to the Greek alphabet before. So that's easy enough, because it's probably not going to be the last time you'll see Greek letters, if it is your first time to see Greek letters. All right, I'm not going to claim to be an expert here, but I don't mind taking a stab at it. So when we see, and why am I doing this? Well, we see in prokaryotes, non-membrane bound organelles. We just call it DNA polymerase 1, 2, and 3. In eukaryotes, though, we use the Greek letters to signify the differences between them. So just quickly here, again, it's not prerequisite knowledge. If nobody's taken a moment and explained these to you. This is lowercase alpha. It's roughly a capital alpha. This is alpha. And I don't know the entire Greek alphabet. Some of you might. I don't. But I know some of the major ones. All right. Lowercase beta, capital beta, really not much difference. I always draw a beta that looks just like a B with a little tag at the end, a little tail. Okay. That way I know it's a beta and not a B. Gamma, which when you type it out, it often looks like a Y. When I draw a lowercase gamma, it looks like an alpha that's 90 degrees rotated. That's how I draw. Okay. Delta. Most of us are probably familiar with capital delta. Lowercase delta is just kind of this curly looking D. Okay. Most of us are probably familiar with theta. I don't know if you've seen theta before, and maybe some of you have seen phi or phi, however you pronounce it. Omega is the last letter. And I'm not going to pretend to, no, you can't see that one. I'm not going to pretend to know the lowercase and uppercase versions of those. But your text here, the reason I took a moment here is because your text does talk about, when you look in the text, they talk about in eukaryotic organisms, we use alpha, beta, gamma, delta, and epsilon. Epsilon, didn't write epsilon on our paper here, but epsilon is also a Greek letter. All right, and those are kind of the first 5 letters in the Greek alphabet. Yes, they match English close, right? English has a slightly different origin than most of the Romantic or Greek-based languages. Let's not get into languages. It's another time. Okay. So again, just kind of a quick review. Helicase splits the double strand. I wrote up here on the board some of the main players. Helicase splits the strand, creates the replication fork. DNA polymerase cannot start on its own. So we need to put an RNA primer in place. We use a primase to add that. So we add just a few nucleotides of RNA. This is the replication process. This is what's going on during S phase before mitosis. We have to copy all of the genetic information, all the chromosomes have to be copied. And this is roughly what we understand of the process of how this occurs. So again, helicase splits the strands. There's 2 strands bound together. Helicase splits the hydrogen bonds, put them together, lay down a small RNA primer that DNA polymerase then can build, replicate the DNA strand. So this is going on both sides. So I'm getting now 2 complete copies of double-stranded DNA as opposed to. And then, and I'm not going to try to pronounce the Japanese name of the Ogasaki. I guess I'll try to pronounce the Ogasaki segments on the lagging strands. And then just another enzyme goes through at the end and make sure everything's all joined up. All the 3 to 5s are joined. Again, DNA polymerase can only go one direction. And it can add 5 primes to the previous 3 prime. All right, so that's kind of the basics of DNA replication. One thing, you know, with prokaryotes, again, I think we mentioned this before. In prokaryotes, they just kind of typically have one single chromosome in this nuclear region, nuclear envelope region. It's not really membrane bound like we see in eukaryotes, but in prokaryotes, it's just kind of this circular chromosome, one continuous loop chromosome. But in eukaryotes, because we see during cell division, we see the chromosomes get packed together. So the chromosomes are long linear strands of DNA. It's all wrapped up and tied up in a nice little bubble, literally. So instead of this continuous loop that we see in prokaryotes, eukaryotes have these long chains of DNA. And there's an end point on those long chains of DNA. We call these the telomere. As best we can tell, they are typically a sequence of TTAGGG, repeated over and over again, anywhere from 100 to 1,000 times is what most sources tend to agree. Now, the problem is, remember, DNA polymerase cannot start itself. So when you get to the end of the lagging strand of the telomere, there's this short, very short end that where we can't lay down an RNA primer and then start the DNA polymerase at the end of that primer. So what happens is the telomere ends up just over time, as the cell continues to divide and divide and divide, over time the telomere gets shorter and shorter and shorter on the lagging strand. And because there's nothing for the leading strand to tie onto, it often clips off as well. That's the reason it's, as best we can tell, why it's the same sequence repeated over and over and over again. So it's an evolutionary solution to this fact that the telomeres will get shorter over time. As best we can tell, that's one of the factors of why we age, is that eventually, once those cells divide enough times, that telomere continues to get shorter and shorter. Your text does talk about, I learned this, that there is an enzyme, telomerase, that will actually generate new telomeres. And they find sometimes in cancer cells, because they rapidly reproduce, they eventually will run out of telomeres. Cancer cells often, the only way they survive and continue or remain viable is if they figure out how to turn on the telomerase coating, which normally is deactivated in those cells. It's how we age. So one anti-aging solution that scientists are trying to approach is turning on the enzyme that generates or lengthens the telomeres. If the telomeres don't continuously get shorter, if we can actually regulate and keep them a reasonable length, then in theory those cells can continue to divide indefinitely. Okay. So interesting little bit tidbit. Like I said, that's something as I was reading through your text, I was like, oh, I'm sure I knew that at some point, but I darn sure didn't remember. Okay. But again, telomeres are not necessary in prokaryotes, because it's just a loop, but since in eukaryotes we have chromosomes, those chromosomes have to have endpoints, those are telomeres. Okay, we don't have to go real in depth with DNA repair, we just need to mention that these are molecules and they are being replicated fast, right? Your text says in prokaryotes it happens really fast. Eukaryotes, it goes quickly. I think your text will give you the numbers. It's a fraction of what prokaryotes go, but it still goes quickly, right? Accidents are going to happen. They're not going to be perfect. In general, the DNA polymerase enzymes are pretty good at proofreading and catching their own errors. They're just kind of relying on nucleotides to be floating around them, and they can grab those and put those in the right spot. Sometimes the wrong nucleotide will drift into that enzyme, and it'll put it in place, and it fortunately has mechanisms where it will realize that the wrong enzyme or the wrong nucleotide is there, and it'll eject it and bring the next one in before it moves forward. So it has mechanisms in place to try to prevent it from moving down the strand with the wrong nucleotide in place. But occasionally, it'll put in the right nucleotide, and something will be slightly defective with that particular nucleotide, because there's millions and billions of them. One of them's going to have an error occasionally. And so sometimes they will have an odd shape here, right? You can see here where the two thymines have created a hydrogen bond with each other instead of hydrogen bonding to the other side, and that causes a flex then in the backbone of the DNA. And so after replication has occurred, after the DNA polymerase has made its way down, there will be mechanisms in place where enzymes will go down the strand, look for places that we need to excise the damaged nucleotides and kick those out. And sometimes the DNA polymerase will just accidentally put the wrong thing in here. Remember we talked about purines and pyrimidines. If I put two nucleotides in here and both of them have 2 carbon rings or 2 nitrogen-based rings, they're too big, they'll repel each other, they'll push each other out, and you'll have a bulge here in the strand. So some enzyme will come through and clean up all the mismatches as well. So there's that expectation that you're going to have some errors in there. All right, I've been droning on. You guys tell me. What do we use proteins for? I know, we slept since then. Talked about proteins. What do we use proteins for? 2 examples, at least. What's that?
Create DNA.
Create DNA. What exactly creates DNA? What do we call that? And that's protein.
No.
2 uses that cells, 2 things we use enzymes for. One should be pretty easy. Wait, did I say enzymes or proteins? I said enzymes. I gave you the answer. Enzymes are proteins, right? Enzymes are proteins. That's one thing we use proteins for is enzymes, right? Enzymes are proteins. Another one. Any other examples? It's not all enzymes. There are things we use proteins for. What was the major topic of last exam? Oh, that's actually chapter 12. That's on this exam. So before chromosomes. Well, I mean, chromosomes were involved in the last exam, though, but not traits. That's genetics. We're talking. That's kind of what we're here on this one. This is the focus of last exam. What's that? Cellular division, mitosis and meiosis. All right. How do the chromosomes get? What moves the chromosomes? Mitotic spindle, all right? Mitotic spindle uses these microtubules, those are proteins. Any kind of structural components within the cell. So a lot of times proteins are going to serve structural component usages, in addition to having enzyme action. We also talked about cellular communication. So a lot of times, a lot of the extracellular receptors, you're going to have some protein component. You're going to have protein connecting this part that's outside the cell, the extracellular domain, with the intracellular domain. You're going to have this transmembrane protein structure, tying the 2 together. Structure. Right? Structure and proteins are 2 simple things we can, or sorry, structure and enzymes. In my head, I'll interchange proteins and enzymes sometimes. Structures and enzymes are 2 of some of the most important things we're going to use proteins for in a cell. Okay. All right. What makes proteins? This is 2 exams ago, right? 2 or 3 exams ago. What are proteins made of? Amino acids. Amino acids. Chains of amino acids create proteins. Okay. How do we produce proteins? We've already said that. It's ribosomes. Ribosomes use RNA to create proteins. Well, it's the central dogma, if you will, of biochemistry. Replication. So 3 big terms right here. Replication, transcription, translation. That's kind of the focus of this exam. The last exam we focused on cellular division. This exam, our focus is really on genetics. How are genetics expressed? Well, genetics are transmitted by nucleic acids. It's important for cellular division to occur. We're going to have good copies of those nucleic acids and to pass on to offspring. We're going to have good copies of these nucleic acids. And what do? Well, those nucleic acids allow us to generate proteins. Those proteins determine the phenotype, the genetics that are expressed of the individual. Three big terms here: replication, transcription, translation. This is basically kind of the focus of chapters 13 through 16. The focus is focusing around these three concepts right here. Okay? Chromosomes are what transfers genetic information. Chromosomes have to be copied. That occurs during S phase, during interphase. Before mitosis can occur, you have to have before I can make two brand new cells, I've got to have two copies of everything. So I got to copy all the DNA. That's replication. We just saw a video, wonderful video on YouTube of that. You've got it in your text. We talked about the basics here. Transcription, because the DNA doesn't leave the nucleus. Eukaryotes, DNA doesn't leave the nucleus. So we create RNA, ribonucleic acid. And then we can send that ribonucleic acid, we can send that out into the cell, and the ribosomes pick that up, and they build protein from that. So how does that happen? Well, let's talk a little bit about that. First, let's talk about transcription. Transcription deals with converting DNA to RNA. So we've seen this before, but it's been a while. We were in the other room. We saw this like third or fourth night, maybe. RNA, ribonucleic acid. There are four types of RNA. You got your messenger RNA. That carries the message from the nucleus to the rest of the cell. You've got tRNA, transfer RNA. And we still don't have to necessarily focus on our ribosome DNA or microRNA. We don't necessarily have to focus on either of those. Let's focus on mRNA and tRNA here. Okay. Transcription. We also have RNA polymerase. RNA polymerase. So replication is carried out by a number of enzymes, but one of the biggest ones is DNA polymerase. Transcription is carried out, again, by a number of enzymes, but one of the biggest ones is RNA polymerase. And again, just like all these enzymes, they can only go down the DNA strand in one direction. In the case of the polymerases, they can go from, they can attach new nucleotides in the direction of 5 to 3. They can attach new nucleotide 5' to a 3' of the current nucleotide in the chain. Meaning they've got to go down the DNA strand in the opposite direction. Right? Just like, so we see here, you've got 5' to 3' on this top strand. The strand that's actually being transcribed, transcription's occurring on this bottom strand. And it's going anti-parallel, that's 3 to 5 if the top one's going from 5 to 3. And you see the RNA strand is coming out also 5 to 3, anti-parallel to the DNA strand being copied and transcribed, I should say. Again, the RNA polymerase moves down, talk about the template strand, I'm going to talk about the template encoding strand here in a second. It's going to move down the template strand. It's going to generate something that's it's going to generate RNA, messenger RNA, that's anti-parallel to the template strand. The other strand, again, there's two strands of DNA here. The other strand, we normally don't copy off of it, except for replication. As far as RNA, the RNA normally, as best we can tell, gets copied off of just one strand. We call it the template strand. Again, it can only go in one direction. Now, I think we mentioned this before. I know we just mentioned it earlier. Right? Remember, RNA does not contain thymine that we see in DNA. One of the biggest differences between RNA and DNA that we need to be aware of is RNA uses uracil in place of thymine. Now, the uracil will, just like the thymine, the uracil will bond to the adenine with 2 hydrogen bonds. RNA still uses cytosine and guanine. Both those, they will pair up using 3 hydrogen bonds on opposite sides. Normally, RNA, when it's just floating around and being used, it's single-stranded. It's not like DNA where it's going to move around in 2 strands. RNA is just 1 strand. Now, it will interact with other things. tRNA, for example, we're going to talk about. As I mentioned, you've got a template strand, which we've mentioned already. The template strand is the side of the DNA that's being transcribed. Template strand, this guy here. The coding strand is the other one. Notice GG, again, this is DNA on top, RNA's got uracil there, and you just go down the line. The coding strand is going to contain essentially the same code as the messenger RNA that's getting transcribed. If you go down the line on the RNA strand here, compare it to what's on this on the top coding strand, you'll see they're the same with the exception of uracil and thymine. And they're both in this 5-to-3 orientation. They're anti-parallel to the template strand. Okay. So now, what's this? See, what the heck does any of this mean? All right. This is transcription. And this is how transcription works. DNA convert to RNA. So what do we do with that? Well, now we got to translate it. We have this messenger, the message. We have this RNA, mRNA. It's a copy of the DNA. It's not the DNA. The DNA is the important part, right? Well, kind of. The mRNA, honestly, is the most important part. It's the message, but it has to have a good template to be copied from. So DNA has to be right, or else the mRNA is garbage. And if the mRNA is garbage, we don't have proteins. And the DNA can't leave the nucleus. So the messenger RNA serves as the messenger. The mRNA can leave the nucleus. It's small little snippets. It's not where the DNA is this massive, long strand, continuous strand. So it's not going to leave. We want to keep it trapped. It's important. It doesn't leave the nucleus unless we're dividing the cell up. And then once we get the cell divided up, lock the DNA back down, put it in the safe. But the mRNA can come out. It's kind of like writing a check, I guess, if you think about DNA being the money and the mRNA is the check that we're writing. Right? And the TNR, I guess if we go further with this analogy, I'm not sure we should. TNR, the tRNA is kind of like the dollars if the mRNA is the check. I don't like that for this one. Ignore that. So again, the mRNA, the messenger RNA can leave the nucleus. And it's ready to be read by the ribosomes. So what do the ribosomes do? Well, the ribosomes are going to link up with this mRNA, and they're going to translate it into protein. So the mRNA comes out, ribosome locks onto the strand of mRNA once it leaves the nucleus. That's where the ribosomes are. They're outside the nucleus. Some of them are stuck to the endoplasmic reticulum. So once the ribosomes lock onto this RNA strand, they're going to move down the RNA strand, and they're going to build a protein based on the information contained in the RNA strand. I didn't realize that first one doesn't loop. That's okay. And eventually you'll run into what we call the stop. Right? So they'll build a long enough protein, and once we reach a certain point on the RNA, there's information there that tells the ribosome, release, you're done, let go. Let the protein go, it's either ready to go to work, or it might need to go to the ER, the endoplasmic reticulum, to be bent, folded, repackaged, sent out somewhere else. Depends on the final purpose of that protein. But all the ribosomes do is they make this string. The protein might need to be refolded and reshaped. We talked about protein structures. I know it's been a hot minute. When we talked about protein structures, we said there's like 4 levels of structure. There's the primary structure. That's the order of the amino acids. The primary structure of the protein is the order of the amino acids. That's what the RNA is messaging. That's what it's carrying. It's the order of the protein, the amino acids. The secondary, tertiary, and quaternary structures of the protein, it talks about its shape, its folding, and what it's used. And as we've learned later on, those enzymes, what those enzymes do is based on their shape, they can either twist or stretch or hold molecules together or pull them apart based on the shape of that enzyme. The shape's important, but here we're just talking about the order, right? The order of the components. And then other parts of the cell can bend those proteins as they need to. But here it's really important. The first step is we've got to get the order of those proteins built correctly. And that's what the messenger does. The messenger takes the information from the DNA, gets it where it is something that can actually build the proteins, and says, here's how you build the protein. So how is that written, right? Because you look, I pulled off some information off of this RNA strand, right? This RNA strand is coming out U, A, G, G, U, A, C, G, G, U, A. What is that? How does that help us? What's the language here? The language is called, we call it codons. A codon is a triplet of nucleotides that specifies particular amino acid. So again, the primary structure of the protein is the order of the amino acids. It has to be right. DNA contains the information to build the protein. The RNA carries that information out, and the ribosome puts the protein together using the codons. So codon is a particular sequence of 3 letters on the RNA that the ribosome is going to use to figure out which amino acid fits on at which point in the protein. There are at least 20 amino acids that are important to especially humans, most mammalian forms of life, most animal forms of life, 20 amino acids. We know there are some others. We'd have to go back and review a few chapters ago about what makes an amino acid. There's good information there. I'm not testing you over that at this point, okay? I've already seen that. 20 amino acids. Again, a codon is 3 nucleotides, 3 letters. Okay? There are 4 possible letters on RNA. That's U, A, C, and G. So there's 4 possible letters, and a codon contains 3 of them. So the possible combination is 4 cubed. That's 4 times 4 times 4. It's 4 times 4 is 16 times 4 more to make 64. So there's 64 possible combinations in the codon language. There's only 20 amino acids. So there are repeats. Okay? There's some, this is the codon alphabet, if you will. So again, a codon comes in 3 letters. Kind of like this is it's one of the best presentations I've seen of this. There's a wheel, there's like a circle that works pretty well. But this is kind of similar to like the Punnett squares we talked about. I'm asking you to do one of the homeworks. This is similar to multiplication table. Again, I think it's one of the better presentations. This one's out of your text. So you're categorized together, right? First letter being a U, second letter being a U. So UU, all these are UUs, and then all 4 possible combinations. Okay, and you go to the next block. First letter is a U, second letter is a C, and then all 4 combinations after that. Okay, and again, there's some repeats. There's 64 combinations, and there's only 20 amino acids that's being coded for, so there's going to be some repeats there. The MET is the initiation. That's the start. That's the amino acid in here. This guy, methionine. So that is the initiation. That's the start amino acid. And then when we get one of these 3 combinations, UAA, UAG, UGA, that tells the ribosome, bring in the release mechanism, let go of the protein. Now you can let go of the RNA also and move on to the next RNA and repeat. Rinse and repeat. Ribosomes also work in a particular direction. Again, 5' and 3'. Direction's important. Only go in one direction on these guys. It's just because it's structured differently in the other direction. Think about like a zip tie, like a cable tie. Ever use one of those, right? Put the plastic piece, you put one end in, and you can tighten it down in one direction, but you can't get it to go in the other direction because of the shape. Same thing here. These proteins, these enzymes in the ribosome are shaped to go down these nucleic acids in one direction, not the other. Now, the tRNA, and that's what we're seeing coming in, dropping in and putting additional amino acids on the chain here. So the tRNA, which is the anticodon, it has 3 RNA nucleotides on it. And they'll match up then with the complementary side on the mRNA molecule. And those tRNAs will carry in, they'll bind to an amino acid. So they're just kind of floating around. They've attached to an amino acid. They're floating around the ribosome, these tRNAs are. And the ribosome will pull those in, and the right-shaped one will fit onto the next line in the sequence. And it'll release the previous one, form a peptide bond, so it's going to bring in ATP to generate enough energy, or bring in enough energy to form the peptide bond between the amino acids. All right? And then move on to the next codon. Rinse and repeat. Rinse and repeat. So how does that help us? And that's kind of where we finished up. I ran out of time today, and that's okay. Got a little less than an hour today. Where do we go from there? So chapter 16, we talk about gene expression. Expressing of genes. How is that expressed? Well, depends on what proteins are coded for on the chromosomes. Whatever proteins that you have the information to build, that determines your phenotype. That determines what traits are actually expressed outwardly. Okay? And that's kind of the focus of chapter 16. It's not real long. I looked at it, I'm like, we can finish that up on Monday. Okay? Figured this is pretty dry stuff. I appreciate you guys toughing it out. All right. That's not a bad stopping place. So I'm going to stop here.